My name is Tutor Ivan Seruneli. Welcome to Easy Science International. In this tutorial, we are going to look at a phenomenon known as photorespiration. For those who have subscribed, thank you so much. And please, if you haven't subscribed yet, subscribe to this channel so that you don't miss any of the tutorials we upload on Easy Science International. Share the videos, leave a comment, and we shall always respond to it. Now, photorespiration. In many see-through plants, oxygen and carbon dioxide compete for Rub-P, that is rubellose bisphosphate. So, when oxygen is in high concentration, Rub-P will accept oxygen instead of carbon dioxide. Imagine, define the laws of photosynthesis. Now, in this tutorial, we are going to look at the definition of photorespiration, the reasons as to why it occurs in see-through plants and it doesn't occur in C4 and CAM plants. And then we shall look at its disadvantages and its only advantage. Now, let us consider this equation where Rub-P accepts oxygen catalyzed by this enzyme Rubisco to form for us one molecule of PGA and one molecule of phosphoglycolate. This is the process of photorespiration. Now, a 5-carbon Rub-P molecule yields a 3-carbon PGA molecule and 2-carbon phosphoglycolate molecule. Now, compare this equation 1 with our equation 2, where it is the normal photosynthesis in see-through plants, where Rub-P combines with carbon dioxide catalyzed by Rubisco to form 2 molecules of PGA. Rub-P 5-carbon, carbon dioxide 1 carbon form 3 times 2 molecules of PGA. So you see that this is the normal equation of photosynthesis in see-through plants. Now, instead of forming a second molecule of PGA, we form phosphoglycolate in the first equation, that is photorespiration. Now, the first equation and this process is known as oxidation. Oxidation, because Rub-P accepts oxygen, a reaction catalyzed by an enzyme known as Rub-P oxygenase. Rub-P oxygenase. Now, however, for the second reaction, it is known as carboxylation. Carboxylation. So, here we see that Rub-P accepts carbon dioxide in a reaction that is catalyzed by an enzyme known as Rub-P carboxylase. Rub-P carboxylase. However, we always take this reaction to be catalyzed by one enzyme which we commonly refer to as Rub-P carboxylase oxygenase, that is rubellose bisphosphate. Rubellose bisphosphate carboxylase oxygenase, and that is Rubisco. It has 2 active sites, one for carbon dioxide and one for oxygen. Now, that is leads to what we call photorespiration. Now, this enzyme has a high affinity for oxygen. So when actually it has a low affinity for carbon dioxide. When oxygen is in high concentrations, it will fix oxygen instead of carbon dioxide. Now, we see that the PGA which is formed enters the normal Calvin cycle in the process of photorespiration. It enters the normal Calvin cycle. However, our phosphoglycolate molecule is metabolized to release glycolate. And this process requires energy. Therefore, we remove a phosphate from phosphoglycolate and we form glycolate where we ionize ATP energy. So this process utilizes energy that is formed in the light-dependent state. Now, these glycolate molecules which are formed, they then combine to form for us PGA and carbon dioxide. And this carbon dioxide is evolved. So there is one carbon formed and it is lost, and we form these three carbon compound which enters the normal Calvin cycle to yield carbohydrates. Now, this is what we call photorespiration. And in brief, its definition, it is photorespiration is the light dependent uptake of oxygen and the release of carbon dioxide. In the chloroplasts of C3 plants. So basically, you uptake oxygen and you release carbon dioxide the way it is in respiration. And it is light dependent because it utilizes energy which is obtained from the light dependent stage of photosynthesis. In brief, that is photorespiration. Now, why does photorespiration occur in only C3 plants and doesn't occur in palm plants? Basically, there are two reasons. One, C4 and palm plants use PEP, which is phosphoenolpyruvate, as their initial carbon dioxide acceptor. And this PEP has a higher affinity for carbon dioxide than oxygen. Therefore, even if carbon dioxide is in low concentration, it will never accept oxygen. It will instead accept carbon dioxide and follow this trend where it will form for us oxalic acid and the rest. So that is one reason it won't occur. The second reason is in these C4 and CAM plants, marate accumulates carbon dioxide next to the group P molecules after being decarboxylated, such that these group P molecules will have a higher carbon dioxide concentration next to it, such that it just accepts only carbon dioxide instead of oxygen. So we saw that these C4 and CAM plants have PEP as the initial carbon dioxide acceptor, and it has a lot of high affinity for carbon dioxide. Therefore, it picks this low concentrated carbon dioxide from the atmosphere. It converts, actually it fixes it to form oxaloacetic acid that is reduced to muriate. Now, muriate is decarboxylated and releases the carbon dioxide to concentrate it next to the group P molecules, producing the PGA, which enters the normal carbon cycle and releases carbohydrates. Basically, those are the two reasons as to why photorespiration doesn't occur in CAM plants and C4 plants. Now, what are the disadvantages of photorespiration? One of the disadvantages we can look at is it leads to loss of carbon by evolving carbon dioxide. You see here, we lose this carbon, and we know that carbon is required in formation of the carbohydrates. So we lose it when we evolve it in this process of photorespiration. Another disadvantage is it wastes energy. You see, in fixing carbon dioxide, we do not require energy. We just require enzymes in our normal photosynthesis. We just fix carbon dioxide with group P to form PGA at this stage. However, in photorespiration, we see metabolizing of phosphoglycolate to form glycolate, a process that requires ATP energy. So you incur another ATP, which is wastage. That is the second disadvantage we can look at. Now, another disadvantage is photorespiration leads to what we call a reduced rate of photosynthesis by engaging some of the group P molecules in accepting oxygen. By accepting oxygen, the rate of photosynthesis is reduced because instead of releasing 2 PGA molecules, you are releasing only 1, as opposed to the 2 which can be released from accepting carbon dioxide. The last disadvantage is photorespiration leads to reduced photosynthetic output or primary productivity. So by engaging or by evolving some of the carbon dioxide and only producing 1 PGA at a time, the primary productivity or the quantity of carbohydrates formed is low. Again, what disadvantages we've seen them? What is the advantage of photorespiration? It is only one, and it is photorespiration allows photosynthesis to occur in conditions of low carbon dioxide, and this is done by accepting oxygen to form PGA that enters the normal carbon cycle to release carbohydrates. Basically, that is the only advantage of photorespiration, and we've seen that it is a wasteful process. Now, in our next tutorial, we shall look at the geographical distribution of these plants, C3, C4, and the CAM plants. Don't forget to subscribe to Easy Science International, like and share our videos. Leave a comment. It might be a question, it might be a suggestion. We shall surely respond to that. I remain Tutor Ivan Serujoji. Stay glued to Easy Science International.